Okay, so since we've looked at the biorisk management strategy, now let's look at some of the parts of the strategy a little bit more in detail, especially on your hazard identification, as well as your likelihood and consequence type of determinations, as well as your risk determination. So what are some of the methods you can use to actually identify your hazards in your laboratory as you're doing a risk assessment? So you've done your work activity, or you've described your work activities. Now, what are some of the methods you can use to actually identify your hazards? So this hazard identification is either through identifying your biological agents or any type of hazards that you may have in your work activity process. So one way of identifying hazard or hazard identification is through a literature review. So for example, you may want to review scientific articles about the biological agent that you are actually you actually want to determine how hazardous it may be. So you may want to review some scientific articles. So these articles will tell you more information about the biological agent that you're handling, and this information will now guide you, or they will give you hints on where your likelihood and consequence will fall as you are doing your inherent risk determination. Another thing is you can go online and download some of the pathogen safety datasheets. So the pathogen safety datasheets are simply datasheets that will give you information on a particular biological agent. So take for example, if you are doing a risk assessment on E. coli, you can go online and then check for a pathogen safety datasheet and then find more information on that particular pathogen. So it will give you information on the host ranges of this particular agent. It will give you the effective dose. It will give you the transmission routes. It will give you history on some laboratory acquired infections that have happened before while it's handling this biological agent. So it will give you a lot of information that will tell you, or will guide you, or will help you as you are determining your likelihoods and your consequences in the further steps of the risk assessment process. Also, you can look at guidelines that come from either CDC or WHO. Another thing is you can look at agent identification and classification. So you need to check, okay, what is the risk group for this biological agent? Because the risk group will tell you information about the treatment availability, how infectious it may be, and how long does the infection last. So you need to have this information. Of course, some of this information you get either from your scientific article research or your PSDS type of review. Then another way you can actually identify hazards about a certain work practice or a certain activity that you want to actually do a risk assessment on is doing what we call a process mapping or workflow analysis. So you may be doing a risk assessment on how you test for hepatitis. So the best way to know which part or at which point can you get exposed to hepatitis while it's doing your testing, you need to map out the process of testing for hepatitis. So what is the procedure of actually testing, or what procedure are you using? So you then map out the process from the time the samples are collected for testing for hepatitis up to the results release of hepatitis. Then you look at the gaps in the workflow or in the map. So you look at the gaps. Where in the workflow can you get exposed? to these particular biological agents that you're actually testing. Then through that, you can now close those gaps, because if you close those gaps, it means that now you would have reduced the risk of exposure through those gaps that you would have identified in the workflow. So this is one of the processes, or one of the hazard identification methods we use during our risk assessment exercise that we'll do later on in the sessions. So we'll do mostly the process mapping, and then also we'll do some form of literature review using the PSDS. Then another method is facility and equipment inspection. So as you are doing maybe your process mapping, or as you are doing your literature review, it's always important to also look at the functionality of certain equipment that may prevent the exposure to certain biological agents. For example, you need to check, how is your biosafety cabinet working? Is it functional or is it not functional? Because it being functional or not functional will tell you something about the chances of you getting exposed to whatever it is you are handling inside there. Then also you need to look at the performance. Is it performing the way it's supposed to perform? So if it's functional, is the airflow working the way it's supposed to work? And then if you're using maybe UV lights and everything, is the UV light, is it able to decontaminate the working area of the biosafety cabinet? So you check the functionality of the equipment, then it will tell you something about what is really happening, or what will happen when you start determining either your likelihoods and your consequences. Another thing is you need to look at, or another method you need to adopt, or you may use to actually get your hazard identification, is historical data review. So under historical data review, most of what we're looking at are previous experiences or past incidences or accidents, or any near misses that may or that almost were incidences or accidents. So you look at the incident management, or you look at your logs, you look at your incident investigations. So you check how many incidences have so far happened where people are exposed either to the biological agents that you are actually handling or anything else. So how many times have people been pricked while maybe testing for hepatitis B? Because when they are pricked, it means now the chances of them getting hepatitis becomes higher. And therefore, if the historical data is showing that you've had a lot of people being pricked whilst they are testing for hepatitis, it means that the chances or probability of an exposure is high. Therefore, your likelihood ratings, as you will start determining your likelihoods, will be higher, will be high. So therefore, you need to review your records for your incidences. Another method you can use is through interviews and consultations with lab personnel or subject matter experts. So if maybe you're doing a risk assessment for microbiology section, so the easiest way to get information about certain biological agents that may be under that particular section, you can just go and ask the people that are actually testing for these particular biological agents, and ask them about the properties about these particular biological agents, because they will have the know-how and they will have the knowledge on each and every particular biological agents that they are handling. And through that information, now you have more of an insight of what exactly the likelihood and consequence of exposure to these biological agents may be. Then standard operating procedure review, or SOP review. So this one is related or is similar to process mapping. So all you need to do is check your procedure. So sometimes you need to check whether or not a procedure may lead to people getting exposed. So you check in the procedure at which particular point can people get exposed to these biological agents while it's following this SOP. Then after that, you may find that, okay, maybe by flaming or by dipping certain things while it's doing culture, there is an increased risk. Therefore, you may try to change the SOP and get something which may not really expose people to injury or to the biological agents. So you can get like a sterilizer, not use the flames and everything. So you look at the procedure itself, and then you look at the gaps in the procedure, and through that you close the gaps in the procedure, and therefore people will use a procedure that will have less risk to exposure whilst they're using this particular procedure. Checklist-based assessment. So under checklist-based assessment, all you need to have is like a biosafety audit tool or checklist, which is checking certain areas about the procedure or about the agent, and through the checking or ticking of certain boxes, you'll be able to deduce the likelihood and consequence. Observation of work practices. So sometimes all you need to do is observe the practice of people. So you may have a biosafety cabinet. So the easiest way is just to observe how are people using this biosafety cabinet. So are they putting on PPE while it's using the biosafety cabinet? Are they not? Are they making sure that they switch on all the switches on the biosafety cabinet? So through that, you'll be able to tell, okay, people that are actually using this biosafety cabinet have an increased chance or likelihood of being exposed to what they are working on in the biosafety cabinet because they are not using the biosafety cabinet correctly. Therefore, the practices that you would have observed will tell you something about where the likelihood and consequence will fall. Then sometimes you may just use risk assessment softwares or tools. So you may just feed the software certain information, and then it will tell you something about your hazard and everything. So there are certain tools that have been created by WHO. For example, there's the WHO RUST or risk assessment RUST. So that tool is mostly used to give you an insight on your hazards and how do you do your mitigation. So you may want to go online and search for the WHO RUST or risk assessment RUST, which is mostly used in the preliminary phases of your risk assessment. Yeah, so that's that on hazard identification. So let's look at determining risks. So we are going to look at certain factors that will contribute to either increase the likelihood of exposure to a biological agent or the consequence. So factors affecting likelihood. So while you're doing a biosafety risk assessment, the factors that we'll look at here are what will contribute to the likelihood rating. So they may either reduce or increase the likelihood of exposure to the biological agent whilst you're working in the laboratory. So one of the factors that mostly affects likelihood is the concentration and volume of the biological agents that you are handling or you are analyzing or you are doing a risk assessment on. So the greater the concentration that you are going to be handling in the procedure or as you are doing the testing, the greater the chances or the higher the chances of exposure. Okay, so the more, like if you have a lot of take for example, if you're doing like serology in comparison to culture. So if you're doing serology for, let's say, cholera, So the concentration of the cholera pathogens in the sample for serology, like in the stool sample, the concentration will be less as compared to the concentration if you culture that stool sample, because then what you've done is you've increased the concentration of the cholera pathogens or the cholera agent. Therefore, the concentration in the culture will be more. So if you are doing a procedure using serological tests, the chances of being exposed to cholera will be less. But then if you're doing culture, because you've increased the concentration at that particular point, and you'll find that the chances of getting exposed becomes higher as compared to the serology. The route of exposure. So what about route of exposure? So there are certain routes of exposure, or certain routes through which you can get the infection that will affect or increase the chances of you getting that infection. For example, if the biological agents that you're handling, you can get infected with it through inhalation, the chances of you getting it becomes higher as compared to another biological agent where you can only get it through percutaneous or through contact, because for you to get infected or exposed, you need to prick yourself. Therefore, most of the time, you find that the chances of pricks would be reduced if people are actually following the correct procedures. But then if it's through inhalation, that becomes a bit tricky because aerosols may be generated in any form, and therefore, if aerosols are generated, you find that you inhale those aerosols, and therefore the chances of you getting exposed to these particular biological agents becomes higher through this route. And then another explanation for routes of exposure is that how can I say it? If a biological agent can be, or you can get exposed to it, or you can get infection through different routes, it means that the chances of you getting it increases. So if the agent, you can get it through inhalation, you can get it through percutaneous, you can also get it through ingestion, it means that the likelihood increases, because if you inhale, you get, you ingest, you get, percutaneous, you get. So if an agent has multiple routes of exposure or routes of transmission, it increases the likelihood of you getting exposed to it as compared to another agent which may just have one type of route of exposure transmission. Stability of the agent. So how stable is this agent in the environment? So the longer an agent stays stable in the various surfaces of the environment, the greater the chances of you getting exposed to it, because you will find that maybe that particular agent may be on the table for or can stay viable on the table for like a week. It means that everyone that will touch that table where this organism is will get that particular or will get exposed to that particular agent. So the longer it stays stable in the environment, the more the chances of you getting exposed to it. But then the lesser it stays if the agent dies quickly. For example, if it's not, maybe it may be intracellular in nature. So if it dies quickly on the surfaces of the lab or the lab coat, if maybe after 2 to 3 hours it dies, it means that the chances of you getting it will reduce, because maybe by the time you're getting exposed to that surface, it would have already died, and therefore you may not get the infection from the agent. Aerosol generation potential. So the greater the aerosol generation potential, the greater or the more the chances that you can actually get exposed to this particular biological agent. containment level, and controls. So the greater the risk group, the greater the exposure. So because you may need some form of heightened control measures for it. So the greater containment level it may need, the greater the chances that you can actually get exposed to that particular agent. Work practices and procedures. So what this means is that if you have poor work practices that are mostly safety work practices, it means that you be increasing the chances of you getting exposed to whatever is in the laboratory. Imagine you have an organism, or you are testing for an organism that can remain stable in the environment for like 2 weeks. It means that if you are not disinfecting, or you are not cleaning your benches, or decontaminating your benches, it means that each and every person who will touch that bench without gloves will get contaminated to that particular agent on the bench. So poor work practices and procedure will increase the chances of exposure to the biological agents you're working with. Pathogen characteristics. So under pathogen characteristics, we'll look at this a little bit more in detail in the next slide. So the various characteristics about the pathogen will affect the likelihood, as we will see in the next slide. Training and experience of personnel. So if people are trained on how to handle certain biological agents, it means that they will be aware of the dangers of actually working with these biological agents, and therefore they will be more safety cautious as compared to people who are not trained, because they will not know how to handle these biological agents. So the greater the training, or the more training you have, the more the less chances you get to, the less chances you have to, or the less chances are for you to get exposed to these biological agents, because you've been more safety cautious. But then if you do not have the necessary training, it means that you increase the chances, because you may be careless, because you don't know what you are exposing yourself to. So the lesser training you have, most of the time, the more chances that people will get exposed to biological agents. Environmental factors. So this one is mostly also just similar to the stability in the environment. So there are certain factors that will contribute to this agent becoming more infectious, or the more you get exposed to it through different types of environments, you can increase the chances. So if you can get exposed to it through the lab, or if it goes outside of the lab, you can still get exposed to it. Or if it can also infect or affect animals, it means that you can get exposed to that. So the greater the environmental or exposure it has, or the greater host ranges it can capture, the greater the chances of you getting exposed to it. PPE. So if you're not using PPE, obviously the chances will be higher. But then if you're using PPE correctly, you are reducing the chances of getting exposed to these biological agents. So let's look at the number 7, pathogen characteristics, a bit more in details. So let's look at the infectious dose of the agent. So the infectious dose simply means how little of or how minimal of the pathogen do you need to actually get exposed to, or get infected with the disease if you get exposed to this particular agent. So the lower the infectious dose or you need for you to get the infection, the higher the chances that you can get infected. The higher the infectious dose, that is, you need a lot of the organism for you to get infected, the lower the chances or the likelihood of exposure to this particular biological agent. because if you only need like 2 organisms for you to get infected, it means that if you just get exposed to 2 organisms, then you get infected. So therefore the likelihood decreases. Mode of transmission. So I already touched this in the previous one. So the mode of transmission. So if this particular agent has multiple modes of transmission, it means that the chances of you getting exposed to it will be greater as compared to something which has less modes of transmission, or if it has only 1 or 2 modes of transmission. Survivability in the environment, I think we've already touched this. If it stays longer in the environment, there are more chances. Ability to cause asymptomatic infection. So if the organism can be able to cause asymptomatic infection, it means that you may have it and you will not even know that you have it. And if you don't know that you have it, it means that you can transmit it to other people, because people will not be cautious. They won't really know that you are sick. Therefore, you are going to be spreading it. And therefore, that asymptomatic infection about this agent will spread, and therefore it will like more increase the chances of more people getting the infection, because it has an asymptomatic type of infection. So virulence, how virulent is it? So if you get it, how quickly do you get the infection? So how virulent is it? So the more virulent the pathogen or agent, the greater the chances of you actually getting infected with it. Antimicrobial resistance. So the more this pathogen has antimicrobial resistance, it means that it will persist either on the in someone's body. And if it persists, it means that people will then transmit it to other people. So if there's an increase in antimicrobial resistance to this agent, it increases the likelihood or probability of you transmitting it to other people or other people getting it. The host range. So does this agent, can it only affect animals, or can it affect animals, humans, then can it also affect plants and everything? So if this agent is able to infect animals, then it can also infect humans, then you can also get it maybe from plants. It means that it has multiple host ranges. It has like 3, and therefore you can either get it from an animal. So if you come into contact, let's say with cattle which is infected with it, you get it. You come into contact with a human person who has it, you get it. You eat a plant which has it, you get it. Therefore, the chances will increase with the multiple, or increase the host ranges for this particular agent. So if the agent has less host ranges, it means that it will reduce the chances of you getting infected with this particular agent. All right, let's look at the factors that are affecting the consequences. So what factors are affecting or affect the consequences? So virulence of the agent. So we touched it under likelihood. So virulence is also affecting the consequences. So if a pathogen will cause a disease very quickly after you get infected, it means that the consequences or the manifestation of the disease will be faster, and therefore the consequences will be greater. Therefore, the higher the virulence of the agent, the more the consequence of the disease, or the more the severity of the disease that you have after you get infected or after you get exposed to it. Infectious dose. So if you get multiple, you get exposed on multiple occasions with this particular agent, or if you get exposed to a high concentration or dose of this particular agent, the manifestations of the disease will be faster, and therefore the severity of the disease itself will also be faster. So if you get exposed to a higher dose of an agent or anything, the disease severity will be higher because of the progression of the disease will also be a bit higher as compared to something which you'll be exposed to which has low concentration or low dose. Host immune status. So how healthy are you at the point at which you get exposed to this agent? So if you are immunocompromised at that particular point, it means that even the disease that you get or the infection that you get after being exposed to these particular biological agents will be more severe as compared to someone who's really healthy and not immunocompromised. So how healthy you are at that particular point or how your immune system is will depend or will actually largely contribute to the severity of the disease. Is there treatment or prophylaxis for this particular agent? So if there is treatment for this agent, like if you get infected with this particular agent, let's say E. coli or Staphylococcus, if you get infected, most likely you may just need to take some form of antibiotics because there's treatment to it. So if there's treatment, it means that you can just get the treatment and within a very short period of time, you may get better, maybe 2 to 3 days, you are better and you're back to work again. So if there's treatment, the consequence reduces. But then if there's no treatment, it means that you cannot get the support or you may not get the treatment to actually expedite the treatment or the getting better after you get exposed, and therefore the severity and consequence after exposure will be greater as compared to this other type of agent which has treatment. Prophylaxis, we're talking about vaccine. So if there's a vaccine for this particular agent, either if there's let's say there's a vaccine that you can take such that after you get exposed, the vaccine kicks in, it means that the consequence will be less as compared to an agent which has no vaccine, because how a vaccine works, a vaccine will actually sensitize your immune system to say, okay, this is how this particular agent looks like. So it will like replicate or it will like look like something similar to the particular agent, and therefore your immune system would have already developed some form of memory B cells or antibodies. So if you get exposed to that particular agent in future, or something now which is the actual agent in future, your immune system will remember to say, we've seen this type of infection before, and therefore the memory B cells will regenerate the antibodies and all those immunocells that will attack and kill this particular infection. So having a vaccine available reduces the consequence. But then if there's no vaccine, most likely there will be an increased consequence. Route of exposure. Multiple routes of exposure, of course, to increase the consequences. Or the route of exposure itself, not just multiple. So you find that certain agents that will use the inhalation type of route will go direct to your lungs. And if they go direct to your lungs, the lungs are a major organ. So if your lungs stop working, it means that you stop breathing, and therefore if you stop breathing, you die. So if the route of exposure is through inhalation, you find that mostly this particular type of agents will have a higher consequence, because some of them may even be fatal, because if they go to your lungs, it means that as your blood now is being oxygenated there, the infection is now also going to your blood. And if it's going to your blood, it means now the infection will spread across your body. So the route of exposure will affect the consequence. But then if you find that the route of exposure is maybe through percutaneous, so maybe if you get pricked, so for this now infection or for this agent to spread across your body, it will become, or it will be a bit slower as compared to an agent which maybe use the inhalation route, so it will be a bit slower, and therefore the consequences will not be really felt quickly, and therefore you may even just get some form of treatment and you get better. Co-infection or underlying health conditions. So under this one, if you have underlying conditions, or if you have people that have underlying conditions in the laboratory, so for example, if you have people that have got hypertension, or they've got diabetes, or any terminal illnesses, it's always best to not expose them to certain sections where they'll be handling high risk type of agents, such as maybe MTB, and maybe the other types of infections such as hepatitis. So what happens is that if you have a co-infection, or if you have already have this underlying condition, if you now get infected with another condition, with another bacterial agent, it means that you have two conditions now that your body is supposed to fight, and therefore the severity of the consequence will be greater as compared to someone who has no co-infection or underlying conditions. Duration of exposure. So this is similar to the dose. So the more you get exposed to the agent, the more the consequences will be felt after you get the infection. Age and general health of the exposed individual. So the younger and the older people who have more serious illnesses if they get infected or they get exposed to some of the biological agents, and also your health. So how healthy are you at that particular point when you get exposed? Pathogen replication rate. So if you get exposed to a pathogen, and if it enters your body, and then if it replicates on a slower rate, it means that the infection or the severity of the infection will be less as compared to an agent or a pathogen that replicates really quickly, because it will spread throughout your body very quickly, and therefore the severity and the consequence of the infection will be greater, because there's going to be like a lot of replicants of the particular agent in your body, and therefore your immune system may become overwhelmed to actually fight the multiple types of pathogens in your body. Okay, tissue or organ tropism. So targeted site in the body. So there's certain organisms that will just target your lungs or target your major organs. So if some of these organisms target the major organs, for example, you have Ebola. Ebola will target your major organs, and then it will hemorrhage, or it will break down all these organs, and therefore you're going to die quickly. Sorry to use that word. So the more this agent targets certain major organs, the more the consequence, and therefore the effects will be greater as compared to something less, which is less tropic when it enters your body. All right, so we've looked at the various factors that affect likelihood and consequence a bit in detail. So now in this next part, we'll just look at how do you now determine your likelihood, and how do you interpret your likelihoods and your severity, so that you now have your risk determined. So on the left in the picture is the risk matrix that I introduced earlier on during the session. So the risk matrix will tell you the ratings for your likelihood, and your possible ratings for your likelihood, and your possible ratings for your severity, and therefore it also predict or to give you outputs for your combinations of your likelihood and severity, so that you know where exactly your risk will fall. So if your risk is falling anywhere where it's green, it means that that's a low risk. So if we need to put it, we need to describe it in scores. So anything that is between 1 to 6 is a low. Then blue will mean it's a moderate risk, which is anything between 8 and 10. And then orange, which is a high risk, so anything between 12 and 15 is a high risk. Then red is very high risk, so anything that will fall between 16 to 25.